Good afternoon. My name is Jim Haber. I'm a professor of biology at Brandeis University in Massachusetts. I'm chair of a subcommittee for the Coalition for the Life Sciences that has invited speakers to this Congressional Biomedical Research Caucus. This is its 24th year, which I think makes it the longest running science cafe in the country. I'd like to start by thanking the co-chairs of the caucus, Representative Steve Stivers, Representative Charlie Dent, Representative Jackie Spire, and Representative Rush Holt for their commitment and dedication and ongoing support of this caucus. Today we'll hear from Dr. Donald Ingber of Harvard University. Dr. Ingber will discuss his really revolutionary research and that he and his lab have worked on that has developed ways of replacing animal testing for drugs and toxins by creating, as he says, organs on a chip, and he will tell you what that means. It's really one of the most exciting developments in the biomedical field. Don Ingber is a founding director of the Wyss Institute for Biologically Inspired Engineering at Harvard, and he has many other hats. He turns out also to be the Judah Folkman Professor of Vascular Biology at the Harvard Medical School, and he's Professor of Bioengineering at the Harvard School of Engineering and Applied Sciences. He's authored more than 300 papers. He has authored also 70 patents. He has received numerous honors for his work, including the Holst Medal, the Pritzker Award for Biomedical Engineering Society, and the Department of Defense's Breast Cancer Innovative Award. He's been elected to the Institute of Medicine of the National Academies and is a fellow of the American Institute for Medical and Biological Engineering. And it's my great pleasure to introduce Dr. Ingber.
Thank you. So I really appreciate the chance to share with you some of the work we're doing, which hopefully you'll find interesting. So first off, I am here as the founding director of the Wyss Institute for Biologically Inspired Engineering. This is our logo. We say we're in the process of self-assembling because we're only 4 years old. The Institute was founded to try to basically to spark the next technology wave in bioengineering. We basically took to a look at bioengineering in the past and saw that it was basically taking engineering principles, trying to solve medical problems, and it was transform medicine, heart transplants and pacemakers and stents and so forth. But we were actually tasked with looking to the future, and what we saw is that the boundaries between all the disciplines are breaking down, that we're beginning to understand how nature builds from the bottom up, largely because of NIH funding and other government funding agencies for the last 50 years, and that we actually think that the future is one where we're going to leverage biological principles to develop new engineering innovations. So we sort of flipped it, and that's what we call biologically inspired engineering. Quick aside, in 4 years we've grown to 350 full-time staff and 100,000 square feet of labs filled, 400 patents, 30 corporate alliances, 8 licensing agreements, multiple startups in discussion, and 1 Science or Nature paper every month for the last 52 months with only 14 core faculty. So I invite you to look at it. I'll give you the website at the end. Organs on Chips is one of our major platforms. I actually head this too. And again, we're tasked with doing high-risk research that will be transformative. And the platform I head is called Biomimetic Microsystems, and the idea is to engineer microchips containing living human cells that reconstitute organ-level functions, not cell, not tissue, but organ-level functions for drug screening, diagnostic, toxicology, and therapeutic applications. And the real long-term goal was to accelerate drug development and replace animal testing. And we felt this is one of the biggest challenges, not only scientifically, but economically as well. And that's because the current drug development model is broken. You're probably familiar with this graph of Moore's law in the computer industry, where computer power doubles every 18 months, and the price per chip goes down and down and down. This has driven our economy for the last 30 years. What you probably are not familiar with is what's called Eroom's law, which is Moore's law backwards, which is that the number of medicine invented halves has halved every 9 years since 1950. And the amount of research dollars going into this has increased dramatically. And there's a real major problem which every pharmaceutical knows about, the FDA knows about, and nobody has a way to solve. And one way that the drug development model is broken is that it currently takes more than $2 million to test a single compound. The studies take years to complete. They take years because they require animal studies, which are required for FDA approval. Innumerable animal lives are lost, so there's ethical implications. And then, in many cases, more often than not, the predictions in animals are wrong when they get to human studies. Now, as a scientist, we would never, ever even think of saying that we would use an approach that's based on being wrong more often than not, yet this is how the whole industry works. So as a result, there's a lack of new drugs reaching patients, and there's been a huge effort to search for better lab models that mimic whole human organ responses and physiology. Now, we had a breakthrough in 2010 published in Science that we think changes the paradigm about how one might approach this, and it's called a human breathing lung on a chip. And the idea was that the major functional unit of our lungs is the air sac, or this is known as the alveolar capillary interface. This is where you have gas exchange, where you have airborne particulates and smog. It's where you give aerosol-based drug delivery, where pneumonias, metastasis, inhalation, anesthetics, all occurs across this interface. And it's a relatively simple structure. There's air in the center, right about here, it's white. This is an electron micrograph through this little section through it, cross-section. There's actually one cell that lines it from the lung cell side. There's a porous, flexible matrix of materials called the basement membrane that holds, it's the anchoring scaffold for cells in the body. And on the opposite side of that same matrix is the capillary blood vessel cell called the endothelial cell, and then there's blood. But I was also trained in medicine, and I know respiratory physiology, we know that critical for function is the fact that we breathe, and that each time I breathe in, the alveolus expands, and when I breathe out, it retracts, and that is absolutely critical for function, as is blood flow, which regulates the vascular system. So when we're trying to basically be inspired by nature's design principles, here the principles are that any organ is defined by having more than one tissue type, usually coming together to form an interface, often between a vessel and the tissue cells, the organ cells. And so that's one thing that we had to do to mimic organ function. And in the case of the lung, we had to mimic dynamic flow in the blood vessels as well as cyclic breathing motions. Now to do this, we took a strange approach, which is we leveraged manufacturing techniques from the computer microchip industry. Now this may not seem evident why we would do this, but the fact is that those techniques allow control over feature size scale. That's the same scale that living cells and tissues live at, the nanometer to micrometer scale. And so what we've done, I'm going to show you with a little video. If you're standing close to the speakers, it may be loud, so don't get scared, because it has sound. So I'm going to let it describe how it works, because it does it better than I do.
The lung on a chip is crystal clear, flexible, and about the size of a small computer memory stick. But it contains tiny hollow channels created using microchip fabrication techniques. A porous, flexible membrane separates the two channels at the center of the device. The opposite sides of the membrane are lined by human lung and capillary blood vessel cells. This mimics the arrangement of lung and blood vessel cells in the air sac of the lung. Application of cyclic suction in side channels makes the entire flexible sheet and cells stretch and relax rhythmically, just like our lung cells do when we breathe. In the lung-on-a-chip device, air flows over the top of the human lung cells, and a liquid medium containing human white blood cells flows below the capillary cell layer. To test how well the lung-on-a-chip device replicates the natural responses of living lungs, we introduced bacteria into the air channel to mimic an infection. And we introduced white blood cells to the blood channel. We then saw the white blood cells migrate across the capillary cell layer, through the pores of the central membrane, and into the airspace where they engulfed the bacteria. Bio-inspired microdevices that can mimic whole human organ function, such as the lung on a chip, could potentially replace animal testing and bring new therapies to patients faster and at lower cost in the future.
So what you've just seen is the entire human inflammatory response in real time at high resolution, this little rubber chip. Now, I think people think that the pharmaceutical industry just does a lot of screening of chemicals till they find one that works, but the fact is they need to understand mechanism of action. They need to understand mechanism of toxicity. So this is extremely, extremely important. So we originally were funded by a seed grant from our institute, but we were lucky to get support from NIEHS, National Institute of Environmental Health and Sciences, early on. And they were interested in nanotoxicology. Some of you may be aware that there are nanoparticles in the environment that can get across our lungs, across the blood-brain barrier, seminiferous tubules. No one understands how this works or how you could screen for it. So we worked on the lung. So we began to explore simulants of airborne particulates. And because we could do high-resolution imaging, we can measure things called reactive oxygen species. These, when people say free radicals that are bad, and you want to take things to get rid of free radicals at the health food store, that's what you're worried about. So we can actually quantitate release of these, which basically is indicative of an injury response, in our chips. And we use simulants of these airborne particulates that are 12 nanometers. That's a billionth of a meter. I often describe a nanometer as a nanometer is to a meter, which is about a yard, as the diameter of a blueberry is to the diameter of Earth. And that's actually accurate. So we're talking small. Now, this is a lot of data, but the point is that there are 3 lines here that you don't see any change, and that is giving all of these particles without breathing motions. If you give these with physiological breathing motions, only with physiological breathing do you see injury. This is important because in the lab, we usually put cells on a dish or sometimes we make layers on a porous membrane that are rigid. We would have never seen this response in vitro. And we found that increased breathing also triggers the entire inflammatory response. This red means that the capillary cells get activated to bind to immune cells, and here you could see immune white blood cells, neutrophils, being attracted to the lung only by breathing plus nanoparticles. Breathing alone is perfectly normal. The nanoparticles alone have minimal toxicity, but both together you see toxic response. We could also quantitate uptake of these particles, these little blue dots here being taken up by the cell, but again, much higher with physiological breathing motions. And then we can measure bioavailability, which is the thing that people are worried about, which is absorption of these into the blood circulation from the air. And as you see here, at the bottom, the pictures with all these dots are with breathing motions, we get an 8 to 10-fold increase of absorption than without. This is what flows out of the vessel part of the chip. We deliver these to the airway. Now, no one's ever seen this before. So this isn't mimicking physiology. This is a prediction. So this little chip predicted that breathing is really important for all these things. So we actually went and developed an animal model where we could put a little mouse on a respirator like in the intensive care unit, and you could actually control whether it breathes or not. And you could measure the ins and outs through the bloodstream, and you could deliver nanoparticles, the same nanoparticles, through a nebulizer, and we see exactly the same response. So this little rubber chip, again, not only mimics, but it predicted things no one's ever seen before. Now, we started to talk to pharmaceutical companies who were very excited, but they actually really said our biggest problem are human disease models. So we set out to develop a disease model, and we developed one of pulmonary edema. This is fluid on the lungs. And we used a chemotherapy-induced pulmonary edema. There's a drug called interleukin-2. It's FDA approved for cancer therapy. It works pretty well in certain cancers like kidney cancer, but its major dose-limiting side effect is fluid and blood clots accumulate in the air sacs, in the air space of the air sacs. That's called fluid on the lungs, pulmonary edema. So we took our device, and if you look from above with air, this is what it looks like. And we gave this drug at the same dose that it's used in humans through the vessel. It's given IV, so we gave it IV in our device through the bottom channel. And what we saw over a period of days is that liquid, like a little meniscus, started to accumulate on the sides and then spread and then completely fill the upper chamber. That's exactly the same time course that it's seen in humans at the same dose. And then we actually we put in fluorescent blood clotting proteins, which here you can't see it, but it's flowing. But here you see when you give this drug, you can see blood clot formation in real time. And then if you look by cross-section with confocal microscopy, you see the blood clots, the red, are on top, are in the airspace, even though we added these drugs to the vascular space. This is exactly what's seen in vivo. The neat thing is that the only time that you see blood clots in patients is when basically you have autopsy or a biopsy and you're doing histology. You have to wait months or you wait for death to ever see this. Here we could see in real time and understand what's going on. You also have high-resolution imaging, so you could actually see that this drug induces these breaks in the cell-cell junctions, those black spots. You can actually see the gaps forming between the cells that allows the fluid and the blood proteins to go across. Again, so you could do mechanistic analysis and quantitative analysis, which you couldn't do in an animal, by the way. Again, animals, you give them things and you analyze later by killing them and doing an analysis. So to quantitate this, we gave a little fluorescent molecule that clinicians use to measure kidney clearance of water, which is normally restricted by the barriers of the cells, but if there's a compromise of barrier function, it shifts. So I was traveling in Europe, and the postdoc who did this sent me a graph that ended about here, about 6, and he said, we could see a statistically significant increase with this drug in vascular permeability, which is what we're trying to measure. But there was no breathing motion in that. He then did breathing, and this is what we saw. There was, again, a huge increase with breathing plus the drug. No one's ever seen this before. We went back to that animal model, and we saw exactly the same thing. Now, this is important because there's some patients on ventilators in an intensive care unit who get this drug, and you actually might be able to alleviate some of these effects potentially by modifying the type of therapy that you give. Finally, I worked for 30 years on how mechanical forces regulate biology, that their mechanical forces are as important as genes and chemicals. And we had worked out a signaling pathway that is responsible. It's the fastest thing that changes in a cell when you stretch it, and it's an ion channel. And I was aware that GlaxoSmithKline had been developing an inhibitor of this ion channel. And given that we saw breathing motions are critical for this, we tested this, and we could show that we could completely inhibit this pulmonary edema response in our chip using their drug that's in development. In parallel, they tested this drug in dogs and rabbits and with cardiogenic edema, which is pulmonary edema, which is when the heart failure induces back pressure that causes fluid to go in the lungs, and they saw exactly the same thing. So these were published in Science Translation Medicine last fall as back-to-back sister papers. But the point of this is that this little rubber chip with human cells is showing us drug toxicities, a human disease model, and drug efficacy, which is the beginnings of what the pharmaceutical industry and the FDA need to think about using these types of models as potentially replacing animal testing. Now, we then received a grant it was actually a partnership between NIH and the FDA. FDA decided to give out the first grants, I think in its history, that was called Advancing Regulatory Sciences. This is with the NIH Common Fund. Originally, it's now managed by NCATS. And we were one of 4. And the idea was to link the lung on a chip to another chip that Kit Parker, a professor at the Wyss Institute in Harvard, has developed that takes, down here you can see at the right, there's a little cantilever like a diving board, but it's flexible, and you have living heart cells on it that contract. And when you give them an electrical stimulus to contract when you want, they actually bend. And you could quantitate that, and you could measure contractile stress or force. And you could also use dyes to measure conduction. You could measure electrical conduction like they do for arrhythmias. And so the idea in this was to link our lung on a chip by the vascular channel to the heart to look for absorption and cardiotoxicity of aerosolized drugs and nanotherapeutics. And that's ongoing, and we actually have some exciting results with that. Now, we actually now have, I think, 10 to 14 organs in development in parallel. And I'm going to give you a few examples just to show you the range of what we're finding and that this approach really scales. So last year we published a paper called On a Peristaltic Human Gut on a Chip. So we take cells from the human intestine, an established cell line that's been used for years. To be candid, most pharmaceutical companies use it to measure barrier function in your absorptive barrier function in your intestine, but they think they're terrible cells because they don't look anything like intestinal cells. Yet they use them. There's nothing better. We took the lung on a chip design, but we changed the height of the channel, the width of the channel. Instead of giving breathing-like motions, we give them slower, rhythmic peristalsis-like motions. Instead of giving the high level of flow, we give them a trickling-like flow, like fluid going through your gut. This is the cells at the bottom right being stretched. The way pharmaceutical companies use them, they put them in a little, on a membrane, a porous membrane, so they can measure transport, and they culture them for 3 weeks before they can use them. And they have This white means they're tightly joined. But when you look at them in cross-section, they're flat. They're like skin cells. They don't look anything like intestine, which are shaped like columnar, it's called, vertical. In 3 days with the peristalsis and flow, we get columnar cells. But what was more amazing is as we grow them, we started to see them sort of pile up. And I said to the postdoc, maybe they're forming villi. Our intestine have these little finger-like projections to increase surface area, and that's exactly what we saw. They form little villus projections. They have specialized apical surfaces called brush borders. And this is what it looks like in a movie. This is a human gut on a chip going under peristalsis. If you were to look at your own intestine, it would look very much like this. As someone who's done clinical medicine, it's kind of amazing. And the intestinal villus is a very specialized structure. It has a proliferative crypt at the base, which is where the stem cells live and divide, and then they feed out daughter cells that then move up and become all the different specialized cell types that have specialized function. There are 4 different types. We showed that if we label the proliferative cells with a pulse, so you could basically see where they are for 2 hours, they're at the base. And then we wash that label, and you see where those cells move, and they move up the villus, just like in vivo. And we can then see that markers for all 4 cell lineages are on this. So we've really built a villus in this chip, human. More importantly, you get physiological functions never seen before. So I won't bring you through this, but this is barrier functions, and this is drug metabolism. This is an enzyme, a P450 enzyme that is, the bottom line is these cells, how they've been used for 50 years, they never express it. Give them this peristaltic-like motions, and it's very high levels. They also have glucose reuptake, and they put out mucus, and these cells are known not to produce mucus. Yet we get them to produce mucus at levels never seen before. This is just like in gut. Because we reconstruct this environment, now we can do something that I think is really exciting, which is we could explore whether we could culture microbiome. I don't know if you're aware of what's happening with microbiome, but microbiome are the bacteria that live in our gut and our skin and our nasal passages, and people are finding they're absolutely critical for diseases. I mean, you could literally take an animal with multiple sclerosis, take the bacteria from its gut, give it to a normal, and it gets multiple sclerosis and vice versa, and other diseases as well. If you put microbiome on a cell culture dish, it's called contamination. They just die. So we put them here because we have flow, we have mucus, and, oops, I'm sorry. Let me show you here. And what we find is that the top of this shows you in a control static condition, the black dots mean that you lose barrier function, they die. But at the bottom, you actually get better barrier function when we put a probiotic bacteria that you buy in the health food store that originally came from a human intestine called lactobacillus, like in yogurt. And this is known in humans that probiotics can increase intestinal function and barrier function. And we now actually have, you can see them moving. They actually live in the crypts of the gut, just like they do in the body. Now, I skipped this. This is modern technologies where you do gene microarrays, where you could do 22,000 genes. You could see how their expression changes under different conditions. And the red boxes here at the top left are 4 different static conditions, and you can see they're basically the same, even though we changed some molecules. If we just give them flow, they're totally different cells in terms of what they express, their phenotype. If we give them flow plus peristalsis, they're totally different cells again. So I tell my lab there's no bad cells. They're just like there are no bad kids. They just get into a bad group of friends or a bad environment. And you give them the right environment, you can kind of reboot them. Last week, we published a paper on a human kidney on a chip. It's a kidney proximal tubule. The proximal tubule, kidney is made up into many functional units, and the proximal tubule is where a lot of the drug toxicities occur. And this is a simpler model. We don't yet have distortion or even endothelium, the capillary, but we have a membrane and we have the kidney primary human epithelial cells from the kidney tubule on a membrane. And what we show is if you just give them flow, but flow like in the kidney tubule, not like in a blood system, they become more vertical like they are in the body. They put out enzymes like they do in the body. They have water channels like they do in the body. And they get primary cilia. These are little sort of antennas that every cell has in your kidney, vertical, which you see with flow, but you don't see without flow. And we started to look at functions, and one of the major functions of the tubule is reuptake of protein and glucose. If People who are diabetic, you worry about having protein and glucose in your urine. Well, this is when it's normal, it's being reabsorbed. And we could actually show that these functions are greatly enhanced just by giving them more physiological flow in these chips. And now we're beginning to add other cell types to add greater complexity. And we could also look at toxicity. One of the big problems with toxicity testing is that animals sometimes don't have the molecular targets that mediate toxicities in humans. So you could never see a toxicity in a mouse or a rat, for example. One is cisplatin. Cisplatin is a cancer drug, and it's normally, the toxicity is mediated by a molecule that transports it across the surface, which are different in humans than in mice or rats, for example. And in culture, if we put these cells in culture and you give cisplatin and you measure toxicity, and I won't go into how you measure it, you actually see toxicity even under a rigid dish. But if you try to see the specific toxicity, the one that mediates problems in humans, by using an inhibitor of that pathway, all the toxicity is nonspecific, or let's say 80% of it's nonspecific. But when we put flow, basically it's all specific. So this is a subtle technical thing, but for the pharmaceutical industry and to begin to identify toxicities is extremely important. And the last thing I'm going to show is called a bone marrow on a chip. And this is in review. It's not yet published. It's a totally different approach, but it's kind of amazing also. We've only done this in mouse, but we're beginning to move it towards human as well. The idea here is that the bone marrow is a very complex structure. We use the term niche in that the environment of the bone and the other cells around it are critical to maintain the stem cells that create all the cells of our hematopoietic system, our blood system. And so we took advantage of a technique of tissue engineering where people have shown that you could form whole bone inside the body of an animal by delivering molecules that are called bone morphogenetic proteins, BMPs, and demineralized bone powder. I won't go into how it works, but the net sum is you put these materials in the body, people have shown for years that you form new bone. No cells are transported, just materials. They induce endogenous cells in your body to be recruited and to form bone. And people years ago said that it looked like they form marrow. And so we went to the literature. They do form marrow, but it's not very functional. We went back, we modified it so we can get functional marrow in vivo, and we formed them in a little rubber device that shapes it in the form of a disc so that if we form the bone, and if it does have marrow, we could plunk it out of the animal, put it in a microfluidic device that has the same little hole. So we take the plug and cylinder, put it in, perfuse it, puncture it and perfuse it, and keep it alive in vitro. And so, and if this worked, we could take one bone marrow transplant and make many. We could have in vitro blood cell manufacturing. We could have basically bone marrow models to do drug toxicities or efficacy. So this is a cross-section of that little plug. The orange is bone. This is actually an imaging of the bony material inside it. It looks like a cross-section of a vertebrae in this mouse. And this is at high power. It looks exactly like bone marrow. For those of you who have ever seen a bone marrow, that's what the bone marrow looks like, but it's engineered. So we take this out, and we isolate the cells, and we do sorting of the cells to identify what the cell types are. I won't go into the method, but just focus at the bottom right. This is the distribution of hematopoietic stem cells. Hematopoietic stem cells are the cells that if you just inject these, they can repopulate all blood cells in another patient or another animal. And these are the hardest ones to keep going in culture, extremely hard. So if we look at the blood of the mouse, which is the right bar, and look at the mouse's own bone marrow, which is the second from the right, they're very different because there are no hematopoietic stem cells in peripheral blood. The two left bars are the engineered marrow at 4 and 8 weeks in the animal. And you can see by 8 weeks, they're absolutely identical. So we have the same stem cells there. By the next cells that the stem cells form are called progenitor cells. When they differentiate, we have the exact same distribution of those. Those cells then differentiate to form red blood cells and white blood cells. They're absolutely identical. And then we looked at all the different types of white cells, lymphocytes, monocytes, they're absolutely identical. So we then cultured these, as I said, in our device. And we could see that if we use the standard in the art is culturing bone marrow on a feeder layer of connective tissue cells, they change their distribution. They don't look anything like the marrow from in vivo on the left. In our little device, they look just like they do in vivo for up to a week in culture. So we're keeping this marrow alive. To show it's really marrow, we actually did a bone marrow transplant in a mouse where we lethally irradiated so it would die because it has no blood system. And at 6 weeks, we completely reconstitute it. But to show it's really, you've got hematopoietic stem cells, you have to do 16 weeks, and we saw exactly the same thing. So we now could use this to basically connect fluidically and feed immune cells to other organs. So to end, as I said, we have many organs in parallel, and the long-term vision is what we call basically a virtual human body on a chip. The idea is that we could link different organs through these vascular channels, just like our organs are linked, and you might have an aerosol-based drug that goes into the lung. You see how it's metabolized by the liver and peed out by the kidney and see whether it induces heart toxicity. You might give another one orally to the gut and do the same sort of thing. And we recently got one of 2 grants that DARPA, the Defense Advanced Research Projects Agency, gave out explicitly to build automated instruments to do this, because pharmaceutical companies and FDA are never going to do this like I do with engineering grad students and postdocs. They want to have like a DVD player that you put a DVD in with 1 chip or 10 chips in different order, and they link together and you get analysis out. And so we are building what we call an interrogator instrument that would do this sort of thing. There'd be an organ chip. It would be in a universal chip DVD-like cartridge, and it would plug into this machine. And we've only got funding for about 8 months now, but we have made major progress where we built chip holders, and we've actually started to build machines. This is in our incubator, and there are 4 of these chip holders, and there's a microscope that moves along it. You could actually run the microscope off your iPhone. You can move it around and look at your cells off your iPhone at home. Here's the chips. And this is a big pump. There are IV bottles on the right that has the medium, like in the hospital. This pump in the last 3 weeks has been replaced, so that's gone, and the pumps are these little black things over here now. So it goes right IV bottle to these things with this automated instrument. It's just the beginning. This doesn't have all kinds of sophisticated capabilities yet, analytical capabilities. There's major challenges with different pressures and volumes and different organ sizes. But the feasibility is there, and we're extremely excited, and we're getting. It's been a unique alliance between FDA, NIH, DARPA, academia, pharmaceutical companies, because you got to do this because they know what the problems are, and it's really helped us to focus. So with that, I will end, and I invite you to our website of the Wyss Institute. We actually won the Webby this year, which is the Academy Award of the Internet. We won it for science. We beat out Wired Magazine and NASA Space Propulsion Lab, and I think you'll enjoy it. So with that, I'll end, and I'm happy to answer questions. Thank you. Any questions? Yes?
You talk about the cancer on the chip.
The cancer on a chip. So if you talk to oncologists right now, one of the big things for them, and it's something I worked on since I was a graduate student, is the microenvironment, that you could have the same drug with the same cancer in the same patient, but you have different response at one site versus another, like at the metastasis versus a primary, and a bone marrow versus an organ. And so we think we might be able to begin to explore that, both by making chips that we can have a vascular system, lymphatics and tumors, as well as tumor cells, see if they actually move and metastasize to organs and place them within these different organs. So I think that's really an exciting side. The other thing I didn't mention is that when we first started this, we thought about personalized medicine, so I could have your liver on a chip and your lung on a chip to do drugs for you. And it's plausible, but it's extremely expensive in the current world. But what we are thinking about is what we call sort of clinical trials on chips. Pharmaceutical companies now will often have a drug, spend $500 million, go to clinical trials, fail, take a team of statisticians, analyze the data, and find that, well, actually, there was one small genetic subgroup that responded. And then 3 years later, they do a small trial, and they actually get some success. So we think George Church is a leader in the Personalized Genome Project. He's also at the Institute. He's collected 16,000 skin biopsies from 16,000 humans. They're doing complete genome on every one, and they're doing iPS cells, which are called inducible pluripotent stem cells, which means you can make stem-like cells from each individual that could become liver, lung, kidney, and you could populate chips. And so if you could do analysis of their genome and you could select out for genetic subpopulations that you suspect might have, let's say, toxicity problems or disease problems, we could, in theory, work with pharma to develop drugs for that particular group and be less costly and have a higher likelihood of success. I think that's a real paradigm shifting approach if we can make it work. Yes?
Do you measure results sort of at static time periods or from the chip?
We could do dynamic. We do dynamic. So like, one of the critical things we do that you can't do generally is we have an airway on a chip now where we give viral types of stimuli, and you can measure cytokine cascades, and you can measure recruitment of immune cells like monocytes over time. So they're flowing by, and you could see the dynamics of when they come in or go out. So we have, NASA keeps cells alive in space by having perfusion systems. And so we have complete control over that. So you could be sampling, or you could be adding drugs. You could control the dynamics of the drug addition. So that's the nice part of it. It's not like remembering to come in late at night to feed your culture dish, which is what we do in the lab.
Are there sensors in the chip?
So the DARPA project would be integrating sensors developed by John Wixwell's group at Vanderbilt that are microclinical analyzers, so you'd be constantly measuring glucose, oxygen, lactate, pH, and then that could be built on over time. We're building to take samples from mass spec, like pharmaceutical companies want to do, to analyze drug metabolism and different levels in the blood. And then we have a group at a company called CFDRC that's doing computational modeling, so we could develop models that do PKPD, which is pharmacokinetic pharmacodynamic modeling, which is what they do in animals in pharma, to look at clearance and efficacy and dose, and see if we could translate from this to humans. That's really the project, actually.
Yeah. You're talking a lot about it for diagnostics, but is there any pathway to actually making organs for replacement organs instead of transplants?
So right now, we're trying to deal with, I like to say it's sort of a minimal 3D cross-section through a living organ to be able to look at functionality, to look at critical interfaces, to do analytical stuff. You need mass, pure mass, to replace a liver or a lung. That said, I think in the future, as this matures, we probably will start moving in that direction. There are people who are starting to look at systems like this that are degradable, that have blood vessel supplies, which is critical for any organ engineering. So it might be that you engineer the vascular system this way, and then you add cells another way later on. So things like that are being talked about. There are groups that are doing this extra outside the body for like oxygenation of the lung. But the way we've designed these, it's really explicitly to replace animal studies and to replace in vitro studies, because if, the major shift in biomedical research is when you could culture cells. That just changed it all. But right now we culture mouse cells and rat cells and human cells on a dish where we know they don't look anything like in vivo. If everybody could do this in laboratories, why not? So we think it's critical for basic research as well.
Yeah.
You mentioned NIH funding, NCATS funding, FDA funding, DARPA funding. How is the current fiscal environment affecting your work? Is it limiting what you could do?
So this is one area where there is this strange, it was such a desperate need for such a long time that when our paper came out in 2010, I think everyone shook their heads and said, well, maybe this is possible. And DARPA gave put a huge amount of money to work. And then as soon as they were starting the application process, NIH and FDA aligned with them and put additional money to work. So I think there are two groups funded by DARPA at a large level of funding, and then there are smaller level of funding. I think NIH is funding 17 groups, something like that. So right now, this is not a big field. That's actually, there's been a lot of money put into it. Now, whether that will continue, I don't know. Now, that said, there's money and there's money in government funding, as you all know. So I will honestly say that my grants that were regular R01s that involved this didn't get funded for continuation. So that's the state of the art of how government funding works. It depends on whether things are a focus right now or not. Now, I think you need to have to always link fundamental and applied, because you can go far ahead of yourself one way or the other. But so right now, the funding in the basic side is terrible. And so we're actually losing young people. That's a big concern for all of us. That affects all research. But for these organs on chips, the last year, there was a large amount of money. I don't know if it'll continue.
It's not affected by the sequester law?
I think there may be some cutback by the sequester. But that was funded already, so it's not like new grants coming in. And I think we've yet to be told what it would be, what the impact is for all of that. Yeah.
Any ideas on, from your standpoint, of how to encourage high-risk, high-reward research? Should the federal government be, they need to be good stewards of taxpayer money, so I understand that, but in terms of rewarding or being in the business of giving out grants, for example, the private institutions like the Howard Hughes Medical Institute, Fairfield, biomedical industry, they're all about high risk, high reward, and since they're private, they can kind of get away with it. But any ideas for?
Well, the whole reason we formed the institute is because I don't think any of those groups are doing high risk research. They sometimes give people a lot of creative freedom, but most work that's given to individuals is incremental. There may be great people doing things incrementally, and there may be not so great people. But the challenge we have to deal with now are so interdisciplinary that you have to get people to work together collectively. And you also have to get people to span academia and industry, because we could, no matter how much you do in the lab, it will have no impact on man unless it gets commercialized. And so our institute was explicitly formed to develop a new model for what we say innovation, collaboration, and technology translation. So our measures of success include IP portfolio, corporate alliances, licensing agreements, new startups, and products in the pipeline in 5 years. And we've done all of that, and in 4 years. And so I think there has to be new models. I don't think, to be candid, we could have done it. We started with a large philanthropic gift, and that is the type of freedom we needed to get going. That said, I think there are elements of all the government agencies that are getting more focused on innovative, risk-taking stuff. It's a small, and I think you always need some part of your investment to be more risk-taking. That said, if your whole infrastructure and your ability to train people doesn't go with that, you don't have a pipeline for the future. And I think in the time of international competitiveness, people really have to think about that. We're structured to leverage the whole Boston-Cambridge region. So we have people from BU, Tufts, UMass Worcester, not just Harvard, all the hospitals, which are separate institutes, because you can't do it on your own anymore. And so DARPA, their task is to sort of take risks, right? They have to fail, or else they're not doing their job. I think other agencies are trying to mimic that, but it's hard because I think they don't have that structure that, in my experience, they're not built that way. We need to build in academia new models. And the academic institutions need to see that too.
They're not built that way. You're saying that the thinking of the cultures?
The cultures, the size of administration versus funding, so at DARPA there's a much smaller administration, much higher funding ratio, and people are put to money to work. The peer review processes are different, and also they're tasked with taking risks to have transformative change. I think NIH, in general, my experience is it's been more focused on rigor and making sure things are the best quality work, which is great, but you have to diversify in any sort of portfolio in a time when times that are really competitive, I think. We could talk offline, because obviously I've done a lot of thinking about this. We spent a lot of time structuring ourselves to develop a new path. Okay, thank you.